Hello, and welcome to the ACR Journals on Air podcast. I'm your host, Vicky Shamagam. Why does autoimmunity happen? The immune system is elegant and designed to protect us. So why does it sometimes go awry? The manuscript we will discuss today is Mechanisms of B-Cell Tolerance in Health and Autoimmunity. It was published in Arthritis and Rheumatology, and a link to the manuscript and disclosures can be found in our show notes from today. I'm thrilled to be joined today by Dr. Shaun Jackson to help us answer some of these questions. Dr. Jackson is an attending physician in pediatric nephrology and pediatric rheumatology at Seattle Children's Hospital and a professor of pediatrics at the University of Washington School of Medicine. Originally from South Africa, he earned his medical degree from the University of Cape Town, followed by postdoctoral training in vascular biology at Harvard Medical School. He completed his pediatric residency at Boston Children's Hospital and dual fellowship training in nephrology and rheumatology at Seattle Children's. Dr. Jackson's research focuses on the immune pathogenesis of systemic autoimmune diseases, with a particular emphasis on B-cell tolerance and its breakdown in conditions like systemic lupus erythematosus. His work has advanced our understanding of how B-cells orchestrate early breaches in immune tolerance and contribute to autoantibody production. Welcome to the show, Shaun.
Thanks for the kind introduction and invitation.
So your manuscript begins with a case which at first blush doesn't seem to be directly related to rheumatology. Tell us a little bit about the case.
Yeah, so as you mentioned in the introduction, I did dual training in nephrology and rheumatology. So I wanted to pick a case that I think was both illustrative of some immunology concepts, but also was something that rheumatologists may not realize about or sort of realize that exists as an association. And so the case is of a young adult male who developed end-stage kidney disease from a disease called X-linked Alport syndrome. X-linked Alport syndrome is a disease where you get kidney dysfunction and hearing loss. And for the most part, our treatment is supportive. There's no specific gene therapy for this disease at this stage. And so for this patient, they ended up on dialysis and had a kidney transplant, which is the way we would try and treat patients who have Alport syndrome renal failure. And initially, the transplant was doing very well, and about a year after the transplant, the patient developed with new onset of hematuria and proteinuria and had a rapidly deteriorating decline in the function of the kidney allograft. And so as nephrologists, what do we do for that is we do a kidney biopsy, in most part to see, is this patient having a rejection episode? Are they having acute cellular rejection? But in this case, when you looked at the pathology, what was noted is the patient had IgG deposits along the glomerular basement membrane, essentially anti-GBM disease, which is a rheumatologic disease. So the question is, why this striking co-occurrence? Because these two syndromes, Alport syndrome and anti-GBM disease, are both very rare diseases. If you multiply the risks together, it's sort of one in many tens or hundreds of millions of chance that a patient would have both of these conditions. So the question is, why did this happen? Why did a patient with a genetic disorder develop an autoimmune disorder in a transplanted kidney?
Great introduction. You've given us a good teaser. Now, to understand the immunology of this patient's case, we need to dig into the fundamental drivers of autoimmunity and how tolerance develops. So walk us through some of the basic immunology about B-cell tolerance.
Yeah, so if you go to that point, what's happened in this patient is they have made an autoantibody against a part of the body, the glomerular basement membrane. And so you have to think about, well, what stops us from making autoantibodies? And at first blush, you would think that the risk of autoimmunity is going to be very, very high. And the reason for that is B cells develop in the bone marrow from hematopoietic stem cells. And in order to become a B cell, you have to make what's known as a B cell receptor, which is the receptor on the cell that responds to antigens that individual B cell would see. And in order to make a very diverse B cell repertoire, the B cell receptor is developed by rearranging these gene segments on the heavy and the light chain. So on the heavy chain, there's the V, D, and J segments, and on the light chain, there's the V and the J segment. So this process happens at random, and that's important because it allows any B cell to respond to any potential pathogen that could be in the environment. The problem with that is almost by definition, if something happens randomly, you're going to make B cells that are react to self, that are self-reactive. And we now know that if you go in and clone individual B cells in the bone marrow, you'll see that before they've exited the bone marrow, you'll see that somewhere between 50 and 70%, 75% of B cells are actually autoreactive in the bone marrow. And so that's the problem that the immune system has to deal with. You want to be able to make a B cell against anything, but by making a B cell that can bind to everything, you inevitably are going to create B cells that respond to self.
Great intro. So central tolerance involves both deleting some of those clones and then editing some of those receptors. Can you tell us about those two mechanisms? Why do we have two mechanisms to eliminate these autoreactive B cells, and give us a bit of that background?
Yeah. Yeah. So I just mentioned that if you look at those initial B cells, about half or three-quarters of them are going to be autoreactive. And so that would be a huge problem. We would all develop autoimmune disease if those cells were allowed to exit the bone marrow. And so there are multiple mechanisms that can eliminate those cells. And broadly, we term all of these mechanisms central tolerance. And so the first mechanism is something called clonal deletion, which is if a B cell develops in the bone marrow that's highly reactive to some ubiquitous antigen. So it's reactive to a cell surface protein that's expressed on all cells in the body. That cell will be deleted by apoptosis. So those cells will just be removed in the bone marrow. But there are actually some other mechanisms whereby the immune system tries to salvage B cells that are autoreactive. And the mechanism that you referred to is what's known as receptor editing. And in that case, what happens is because the B cell has both a heavy and a light chain, and on the light chain has two different loci that can recombine, the kappa locus and the lambda locus, what happens is that a cell that maybe isn't as autoreactive that it's initially clonally deleted can cycle through the various different light chain options in a process called receptor editing to try and salvage that B cell receptor, to basically make it less autoreactive and allow it to exit the bone marrow. And you might say, well, why does the immune system go through this effort? Why not just eliminate all of them and allow other cells to get through? And I think the reason for that is just because it's very energy intensive to make all of these cells. And so having a way to salvage and sort of restore a B cell that's moderately autoreactive has a benefit to the immune system to provide that broad repertoire that can bind everything. And so that's really the major mechanisms that we think about of central tolerance. And that covers basically half of how we think about B cell tolerance.
Yeah, so even central tolerance mechanisms are not fully effective at eliminating all autoreactive B cells, as you mentioned. So we also have in the human immune system some peripheral tolerance mechanisms. Tell us about those.
Yeah. So the first thing to state, this is something that I was somewhat surprised about or didn't think about very carefully when I started studying B cell immunology as a fellow, is a lot of us, I think we think of an antibody as being highly specific. It binds one thing. And certainly rheumatologists think about that, right? If we have a TNF inhibitor, that binds TNF. It doesn't bind something else. Our antibodies against flu, COVID, etc., are all very specific to that pathogen. However, in all of those cases, those are B cells that have gone through an immune response and we've selected them to be highly specific, whether it's in the natural immune system against an infection or in the lab at a pharmaceutical company that's choosing a great clone. In reality, though, if you look at a naive B cell, one that hasn't gone through an immune response, it's actually very polyreactive. It binds a lot of different antigens. And so when we think about B cell tolerance, we have to think about tolerance across a spectrum. B cells have different reactivities to different spectrums. And so in the periphery, what happens is clonal deletion deletes all of the immune cells that are highly autoreactive. These are the most autoreactive cells. But then everything that's left, some of them have no autoreactivity and some have a little bit of autoreactivity. And that group of cells exits the bone marrow and then is engaged with a different form of tolerance, which we call peripheral tolerance. And there's different mechanisms that happen, and they happen sort of concurrently. So one of which is what's known as anergy, which is if a B cell responds to an antigen that's ubiquitous but maybe soluble in the body, it will enter a state of tolerance where the cell still exists. It doesn't necessarily die. It has a lower threshold or a higher threshold for activation. It needs a stronger signal to be able to activate and differentiate into a plasma cell. There are other mechanisms as well. As B cells exit the bone marrow and start entering the secondary lymphoid organs, we make a lot more than we need every day. The natural turnover of B cells, we make manyfold more than that. And so there's a competition to enter the follicle. And so cells that are more autoreactive are more likely to be excluded from the secondary lymphoid organs and have a shorter lifespan where they die off. And so that's just a way of thinking about peripheral tolerance as this process that takes intermediately autoreactive cells and makes them less likely to become reactivated by either making them anergic, meaning that they're less likely to respond to an antigen, or by shortening their lifespan or keeping them outside of the follicle.
Yeah, so you bring up the germinal centers and the follicles. Tell us what happens in the germinal centers.
Yeah, so when a B cell, let's say a B cell has made it to the secondary lymphoid organ, what happens there is, let's say you get an infection. A B cell that responds to that antigen will then proliferate and then will actually engage in T cell help and will then go into the germinal center where there's these iterative rounds of selection where B cells cycle between the heavy and the dark zone and the light zone of the germinal center, and cells with higher affinity to the antigen are selected. That's why the antibody that you produce after an infection is a better antibody or higher affinity antibody than it is at the first time you see that response. However, there's a problem that comes up in the germinal center, and it's really the same problem that happens in the bone marrow. So in the bone marrow, B cells are developed by a random recombination of V, D, and J segments. In the germinal center, there is random mutations of the B cell receptor. And so almost by definition, there can be ways that within the germinal center, new autoreactive specificities could develop. So let's say you've got a pathogen that's very similar to a self-antigen. As you're selecting those receptors, you might go awry and start producing a high affinity autoantibody against a self-antigen. Now, the mechanisms, and so for that reason, the immune system has a lot of mechanisms to prevent de novo or new autoreactivity happening in the germinal center. There's a range of sort of germinal center specific tolerance mechanisms. And these are actually quite hard to study. They're certainly quite hard to study in humans, but they have been studied very carefully in a lot of elegant mouse models. And without going too much into the weeds, I would just say that there are ways in which the immune system can highly select in the germinal center antibodies that are specific for a foreign antigen, but don't respond to self. And they can do that at the level of individual amino acids. It's really quite amazing that this happens.
It is amazing. And the immune system never ceases to kind of awe me with its elegance and adaptability. Now, I'm going to take you to a place that you might not want to go now, because I know you're a B cell biologist, but T cells also do play a role in regulating B cell tolerance. Can you tell us a little bit about how they do this and give our listeners some background about the various rare genetic autoimmune conditions that have helped us understand that intersection of how T cells regulate tolerance and autoimmunity?
Everything in science is about hyperspecialization. And so I've focused on B cell immunology and so therefore don't think the T cells are that important, and vice versa. We have T cell immunologists that don't care about B cells. But clearly both are important, right? And they're important in a normal immune response, but they're actually very important in tolerance as well. And so I mentioned before that a lot of low affinity B cells can enter low affinity autoreactive B cells can enter the peripheral compartment. And so the question is, what restrains them from being activated? And one of the things that restrains that is actually eliminating the T cells that are autoreactive to the same antigen. And so we're not going to go into all of the mechanisms of T cell tolerance, but essentially while B cell tolerance is developing in the bone marrow and the periphery, T cell tolerance is developing in the bone marrow and the thymus. And so the most autoreactive T cells are eliminated in the thymus. And what that means is that if there happens to be an autoreactive B cell in the periphery and it gets activated, it won't be able to find the cognate T cell that will promote its activation and promote Bake's intolerance. And this has sort of been elegantly studied in a lot of animal models where, for example, you make a mouse that has a transgenic B cell receptor that binds a particular self-antigen. And what ends up happening is the mouse doesn't necessarily get sick. But when you then adoptively transfer a T cell to the same antigen, well, then you get an autoimmune phenotype. So that shows how the two are linked. And the way that in the review we discussed two rare genetic syndromes which really get at this idea. One is the disease autoimmune polyglandular syndrome type 1, APS1, it's also known as APOSED. So this is a disease where you get mutations in the ARI gene, which is involved in T cell tolerance in the thymus. And then the second disease is a disease called IPEX, an immune dysregulation, polyendocrinopathy, enteropathy, and X-linked syndrome. So this is a disease, it's a rare disease of boys where you have a mutation in FOXP3, which is the master transcription factor for Tregs. And in both of these diseases, you get autoimmunity. And because they're T cell diseases, most people think about these as sort of T cell related autoimmune syndromes. And I think the one thing we wanted to point out in the review is if you actually screen patients with these diseases for autoantibodies, in both diseases, they have high rates of autoantibodies that bind to very sort of defined types of autoantigens. And that to me emphasizes the fact that T cells play an important part in B cell tolerance. If you have a genetic syndrome that impacts T cell tolerance, then you end up with autoantibodies on the B cell side, even though neither of these genes, ARI or FOXP3, are thought to directly impact B cells.
Yeah, it's really helpful to have you explain that and bring it to our understanding. So I have to ask you, from a sort of human evolution standpoint, why is it beneficial for humans to have this incomplete B cell tolerance?
Yeah, so if we go back to that central tolerance idea, I mentioned that high affinity autoreactive B cells are just eliminated in the bone marrow. They just die by what's called clonal deletion. And there's obviously a threshold that's placed in the immune system to only delete the most high affinity autoreactive cells. And so the question is, why has evolution and the immune system just altered that threshold? So think about it another way. You could say instead of eliminating only the highest affinity autoreactive cells, why not eliminate all of them, right? Why not eliminate every single B cell that reacts to a self-antigen anyway? And I think the reason why this doesn't happen, the answer to this paradox, is really the goal of the immune system of B cell development is to create a naive B cell receptor that's very, very diverse so it could respond to any pathogen that the patient potentially saw. And so if you think back to the beginning of the SARS-CoV-2 pandemic, like this was an entirely new pathogen. Nobody in the human population had seen this pathogen, but I was entirely confident that we would be able to respond to this virus and that we would be able to vaccinate against this virus. And that was because I knew that there's enough diversity in the B cell repertoire to respond to any new pathogen. The problem with making too strict a tolerance, if you set your tolerance such that you removed every single autoreactive B cell, that would mean that basically a lot of our diseases that we think about in rheumatology would disappear. We wouldn't get lupus, we wouldn't get rheumatoid arthritis, etc. But what would happen is we'd have these big gaps in the repertoire, and pathogens would exploit that. We would have cases where pathogens would go through the population and kill large numbers of people. And if you think about human evolution, we don't think about this too much because we live in a sort of modern society with sanitation, vaccination, things that protect us from infections. But if you go back a few hundred years, for every child that's born, about 1 in 3 or 1 in 2 would die before the age of 5, and they would die of infection. And so there's a huge pressure from evolution to basically make our immune system be very diverse and able to respond to a lot of things because we just, from the day we're born, are exposed to all of these pathogens that could shorten our lifespan.
Yeah, amazing. So let's get back to the case that you presented at the beginning of this discussion. You mentioned Alport syndrome is a genetic disorder, and it's characterized by abnormalities in type 4 collagen, which is found in the basement membrane of the kidney, the eye, the cochlea. And so these patients present with progressive glomerular disease, sensory neural hearing loss, and ocular abnormalities. Why are they also at risk of Goodpasture syndrome or anti-GBM disease after a transplant?
Yeah, so if we go back to the sort of first principles of B cell tolerance, is that the immune system is tolerized against antigens that they are going to see, self-antigens that the B cells are going to see during development, either in the bone marrow or in the periphery. This disease is unusual. So there's various forms of Alport's disease, Alport syndrome, but the most common is the X-linked version. And that's caused by mutations in a specific component of collagen type 4, if you have that mutation. And so this patient with this disease would essentially be born without that protein. And so because they lack the protein, and the protein is expressed in kidneys and the ear, as well as the eye, they can have some problems that develop, including progressive kidney dysfunction and progressive hearing loss. When the patient has a transplant, that transplant will have that protein. Even if it was a living donor transplant, this would be from a relative who doesn't have Alport syndrome. The patient would be transplanted. And now, for the first time, that patient is going to be exposed to collagen 4, to particular collagen 4 epitopes that they don't have. And this autoimmune response, well, it's actually not technically an autoimmune response, it's an alloimmune response, right? Does not happen in every patient, probably because collagen 4 is hidden within the glomerular basement membrane and is not often seen by the immune system. But the patient is at risk that if something happens such that the immune system now responds to that antigen that is seen as not self because it's never been seen before by the immune system, the patient can have an immune response where they will now make antibodies against the glomerular basement membrane, and they get an anti-GBM disease. And so it's not one in tens of millions of cases that the patient has a rare genetic syndrome and a rare autoimmune disease. They're now at markedly increased risk because the normal tolerance mechanisms that would normally have been developed haven't happened because the patient hasn't previously seen that antigen. Sort of one other interesting sort of vignette about this case is the ways that we test for anti-GBM disease, besides biopsies, is looking for the antibodies against various antigens. And different companies make different tests in different ways. But the classic Goodpasture's antigen is actually not the same component of collagen 4 that's mutated in Alport syndrome. And so there's even been cases where the patient's testing for anti-GBM disease fails because the autoantibody that develops in Alport syndrome transplant is so specific, it's actually going to a different subunit of collagen 4, and so may not be picked up on that test, which I think, once again, just highlights this sort of an amazing feature of the immune system, that we can develop tolerance to all of these different proteins in our body, but that creates a problem that if a new protein comes in, we can then develop an autoimmune response to it.
Thank you for sharing that tip, because I definitely was unaware of that. And that's very important clinically if you're seeing these patients. So I guess there are a few other examples of neoantigens later in life triggering breakdown in immune tolerance and causing autoimmunity. Could you talk a little bit about those?
I think I mentioned in the review, the question then becomes, okay, if all these tolerance mechanisms exist, why do any of us get autoimmune disease in the first place? And I think the one answer is, as I mentioned, low affinity autoreactive B cells persist in the repertoire. All of us, if we went and cloned our B cell receptor and made antibodies from our naive B cells, we would have autoreactive B cells in our periphery. And so the question is what causes that break in tolerance? And I think there were two main things, one of which is genetic polymorphisms in signaling molecules that exist in B cells, T cells, other parts of the immune system. And I sort of highlight a few of those in the review, but really they've been explored by a lot of other review articles. And so I'd rather just decided to reference the genetics there. But basically, the one way is you can have a genetic mutation which alters your signaling threshold. Now these low affinity cells are maybe more likely to be more likely to be activated in the periphery. But for the purpose of this review, I wanted readers to think about another potential mechanism, and it kind of links to that Alport's case. The reason why the patient can develop autoantibodies is they're seeing something new that they haven't seen before in previous stages in their life. And so the question is, are there any other examples of seeing something new, seeing something that the immune system has not been tolerized to? And so there we think about neoantigens. And so I go through a couple of examples of neoantigens that can promote autoimmune syndromes. One of the sort of most widely known is the process of citrullinatation. So if we think about rheumatoid arthritis, that is an autoimmune response against a self-antigen, but it's a modified self. It's post-translational modification by the process of citrullinatation, which promotes that break intolerance. And there's actually a lot of other examples in the immune system, various other autoimmune syndromes, where we think that actually the initial break intolerance might be because, for various reasons, there is a new antigen that's being formed by a post-translational modification or a whole range of other different examples that have come up.
Yeah, that's such a good point to make. And the other case where we see this is, of course, paraneoplastic autoimmune syndromes. Is there a similar mechanism at play there?
Yes, the way to think about this is a cancer is a cell that has been genetically modified. And so for that reason, that genetically modified cell could express an abnormal protein or express a protein in a different place. And so I sort of look through examples of this. And so one of the examples is Lambert-Eaton myasthenic syndrome, which can be a paraneoplastic syndrome. And in that case, that's actually caused by ectopic expression of an antigen that's usually expressed in a very immune privileged site, the eye, the brain, etc., where the immune system wouldn't usually see that antigen, but because the cancer is dysregulated, it starts expressing that protein. And then another example is the link that we've known about for many years about the temporal links between cancer and various autoimmune syndromes, so dermatomyositis and scleroderma. And so in scleroderma, there's a very well-characterized case, sort of association, where patients with mutations in an RNA polymerase type 3 subunit, which you can identify these mutations in the cancer itself, they can develop antibodies against the cancer, which then cross-react and cause a subsequent development of sort of fibrosis and tissue damage characteristic of scleroderma. And so the thought there is that the cancer develops first, exposes a new antigen to the immune system, which then initiates an autoimmune response, causing an autoreactive response in a disease like scleroderma and presumably dermatomyositis as well.
Yeah, no, I think that's really helpful to share, and kind of fascinated to get this deep dive into the immunology of all of these syndromes. I'm wondering, will you speculate a little bit about looking into the future? We've learned a lot about the immune system in the last 60, 70 years. How do you see these insights into immunology shaping the future of autoimmune disease research and therapy?
Basically, it's sort of asking, well, what have I done over the last 10 years or so since I finished fellowship? And really what we were doing in the lab was studying mechanisms of how B cells break tolerance, doing it in a sort of very careful way of saying, is it this gene that's expressed in B cells or that gene that's expressed in B cells? And the way I would sort of summarize 10 years of work is, for many cases, what we did is we took an animal model where we knew that this gene was important in every cell type in the body. So if you knocked out gene X in a mouse model of lupus in every cell, the disease went away. And what we tried to do is say, well, what if you knock out that gene just in B cells? And so we did toll-like receptor signaling, cytokine receptors, a whole range of things. And all of these came back to the answer that B cells are really critically important in the pathogenesis of a disease like lupus and rheumatoid arthritis, etc. And that really implies that we should think about B cell targeting as a therapy for lupus. Now, at the time I started working in the lab, it was very shortly after a series of studies showed that the drug rituximab actually didn't work in lupus, right? There was a failed study in extrarenal lupus. There was a large failed study in lupus nephritis, which showed that rituximab as a B-cell depleting agent didn't work. But on the other hand, we had all this animal modeling showing that really B cells are quite important. So what's the disconnect? And over the years, we recognized that really rituximab was not depleting at the level of depth that we wanted. And so various people around the country and various labs were thinking about, are there better ways to deplete B cells? And there are actually a whole different range. And one of the most exciting recent developments in the B cell field has been CAR T cell therapy.
Yeah.
So this is really a cancer therapy, right, which is designed to treat refractory cancer. After a lot of sort of thinking about how we would design a trial, I think we're very fortunate that a group in Germany, in Erlangen, actually moved ahead and did this trial where they gave CD19 CAR T cells to patients with highly refractory lupus in a way that eliminated all of those cells, didn't just push down the B cell counts, but actually eliminated all of the CD19 positive B cells. And what they found was remarkable is not only did patients get better, but they've entered a state of remission, that they can remain off remission, off therapy for a prolonged period of time. Like, obviously, we weren't the ones that did the initial human studies, but all of this in some ways made sense. The basic research we were doing in animals implied that the cell type was critically important. And now we have the human confirmation that if we go in and eliminate the cells to a deep degree, that we actually eliminate all tissue resident B cells, we can enter a state of tolerance, which shows that B cells are important.
Yeah, so does that mean that B and the T cell biologists have actually started talking to each other then?
Yeah, I think occasionally we have to, right? But that actually raises other interesting questions about why the remission has happened so long, because you would think you've already broken T cell tolerance. So I think this is not the end of research in the field, the fact that we've been able to induce remission in a small number of patients. I think it really comes down to the importance of understanding how this crosstalk between B cells and T cells happen. I can give one little plug because this is a public podcast and I might as well use it to give a plug. But I think we were in some ways ready at our institution, Seattle Children's, to start thinking about using CAR T cells in autoimmunity. And as soon as Europe started showing that this therapy was effective in adults, as a pediatric rheumatologist, we decided that we really needed to study this in children who have some of the more significant or severe cases of lupus. And so about a year ago now, we opened what was at the time the first and only pediatric lupus study studying CAR T cells in the US. And now there's been some other groups around the country that have opened. And so we're now recruiting patients with earlier onset disease to really ask the question, can we restore B cell tolerance? Can we eliminate those pathogenic clones and put the patients back into a state of remission and limit the sort of accumulating effects of medication toxicity that we see in all of our patients? And so that's a really exciting part of sort of moving, for myself personally, moving from animal models of mechanisms to like, can we actually apply this therapy to humans, in particular children with autoimmune disease?
Oh, that's really exciting. And I can see your rheumatology self perhaps putting your nephrology self out of business. Which, good. So I wanted to discuss a little bit more about your career journey. You trained in South Africa. What prompted your move to the US?
Yeah, so I as I mentioned, I finished my medical degree at the University of Cape Town, where we have really excellent clinical training. Perhaps because it's a lower resource country, we don't use expensive diagnostic testing as much. And so I think I was far better with a stethoscope 15 years ago than I am today. But I sort of had this feeling that I wanted to sort of understand more what was going on with diseases and really moving to having a career where I was more focused on the research side of understanding how disease happen rather than just clinical training. And as I'm sure a lot of people recognize, the US is one of the homes for that. The US has made these remarkable advances in science and really performing cutting edge science. And so, like a lot of international students, I looked at ways of moving to a place where I could do what I felt would be cutting edge research. And I was very fortunate. I managed to find a postdoctoral position at a lab in Boston that was affiliated with Harvard University. And in that time, just got my initial exposure to research. And I would say at the time, if my boss at the time is listening, that I don't think I was very effective as a researcher, partially because as I said, I had all of this clinical training at how to palpate a spleen, but had done no experience doing a Western blot. But I, in some ways, just caught the bug. I just felt there was a level of things that I found interesting that when you did an experiment and you sort of developed your blot and you saw something, even if it was hard to explain why this was important to people outside your field, I just found it exciting to be the one person that knew that experimental result on that day, that I was doing something that was sort of at least contributing a tiny part to a little discovery. And so when I caught that bug and decided I wanted to do research, I initially had never planned to stay in the US for residency and fellowship. But I started speaking to people in the local area about, would it be possible to do a residency in the US with the specific goal of becoming a physician scientist? So did all of the exams, the USMLEs, etc., and was fortunate to be accepted to pediatrics residency at Boston Children's, where I said, on the day that I interviewed, that my goal would be in the future to become a subspecialist. I actually didn't know what field I would do. I said I didn't really mind what field it was, but I wanted to have a field where I would specialize in some part of human disease, see those types of patients in clinic 1 or 2 days a week, and then the rest of the time would spend my time studying those patients and working out mechanisms. And I think I've been very fortunate that the US has offered me the opportunity to do that and to continue to do that after fellowship training.
Amazing. So you had a dream and went after it. Tell us a bit more about, you've kind of glossed over the challenges of pediatric residency, which is a long road. You then did dual fellowships in nephrology and rheumatology. What attracted you to those two specialties? And does that mean when you call a consult, you argue with yourself?
Sometimes. You sort of said I had a dream and I sort of was able to pursue it. That has the obvious survivorship bias. So I sort of feel in some ways bad to be even saying it. But, it is kind of true that that's what I wanted to do, right? I was sort of willing to focus on having a research career. That's sort of what I found exciting and was fortunate to be able to do it. It raises an obvious question is why, if I was interested in research, did I choose to do 2 fellowships? That seems to be the opposite of the strategy. And I think the reason I did was I was particularly interested in autoimmune disease, or became interested in it. Even since medical school, I just found a disease like lupus to be fascinating. Like, how could you get one disease that would cause all of these different manifestations in the body? It's not just a kidney disease. It's not just a joint disease. It really can affect multiple parts of the immune system. And so the reason why I chose 2 fellowships was, well, firstly, it didn't double the length of training. You can sort of combine the research portion. So it only increased my training for 1 year, which is, not nothing, but it's not the end of the world. But really the reason I did it was because I wanted that sort of focused clinical expertise that matched with the research expertise. So I thought if I became a nephrologist, I would sort of have to know all of these fields, be a transplant doctor, a dialysis doctor, etc. Similarly for rheumatology, there's a lot of different sort of sub parts of the field. And I felt that if I did this combined training, I could sort of, have this more dedicated focus on autoimmune disease of the kidney, which is what I do. I predominantly see patients with sort of lupus nephritis and autoimmune kidney disease when I see patients in clinic. And then by having that sort of specialty, I would have the time to spend the time doing research for the other 80% of my time, or whatever my grant funding is. So that's why I did the dual fellowships, I could say.
So it's almost like you took the scenic road to the
Yes.
scientist career. So now, looking back on that scenic road, do you have any tips for individuals just starting out in their career on how to establish themselves as a physician scientist and live the dream you live?
I don't know if my wife would say it's a dream, but anyway, I think
She would think you're dreaming, probably.
yes, something like that. Yeah, so I think, the first thing is you kind of have to have a little bit of a thick skin, right? Because I think the way that I think about a physician science career is it's actually kind of fun, right? You get to study things that you find interesting. And the problem with having a fun career is a lot of people want to do it, right? So it's almost by definition, it's always going to be competitive to get grants. As physicians, there's always extra patients to see. And so to become a physician scientist, you need to be able to secure some sort of funding that will buy back your time and allow you to spend enough time in the lab where you can make a contribution, hopefully at the level that somebody who's a PhD scientist would do, right? And so I think my first level advice is the route is long, but it's a fun career over time. But you also have to have a thick skin. I think you always have to say, and you recognize this more after you start out submitting grants, and then as soon as you get your first grant, they ask you to start reviewing other people's grants. And, you start recognizing that when you're sitting on a grant review panel, very few people are submitting bad grants, right? You're really trying to make decisions based on a limited funding source where we don't actually decide who gets funded, but you're really trying to give a score to the sort of value of a grant. And that in some sense is subjective. It's a hard process to do. And so I think my one message would be is even good grants don't get funded, right? There's no scientist that's batting 100 in terms of grant applications. But that's one of the ways that you just have to have a thick skin to be able to continue pursuing this path. The other part of it, which I think is very important for physician scientists, is we do have a lot of clinical training. So if you think about how much time we spend doing clinical training versus how much time you get to do research training in residency or fellowship, you're always going to be a better clinician on the day you graduate than you are going to be a researcher. That's just the natural state of affairs. Maybe if you did a full PhD as an MD PhD, that you could say it's a little bit different. And so one of the things that I think is very easy for physicians is we all recognize the problem, right? We recognize what are the major clinical problems that sort of keep us up at night. What are the diseases that we know we don't have perfect treatments for? So we know the problem. What we don't sometimes know is how to frame a research question to actually get at that problem. How do you because any sort of research study you do is going to take a small bite of that problem, but how do you select that bite of the problem in a way that makes sense, in a way that is a study that can be done feasibly? And so what I would advise everybody to do when you're in fellowship is really lean on mentorship. You need to find researchers that you respect, that you think are doing the job well, and go to them and say, I think it's okay to be pretty broad. Nobody who's a good research mentor is asking you to come up with a hypothesis at that point. They want to have somebody who's broad, who knows generally what they're interested in, and then could present projects that they have that are ongoing or cohorts that they have already collected, where you could probe an individual aspect of that. The goal is never to try and do it yourself. The goal is to find a mentor that can guide you through that process and support you through your training path.
Yeah, I think it's really brilliant advice, and definitely take to heart that advice of having a thick skin. It's a difficult world out there, and any single rejection is not a forever rejection. Just kind of pick yourself up and try again.
Yeah, absolutely.
I want to thank you, Shaun, for sharing this great article with our listeners, which I think will be a classic in the field of rheumatology, for helping us make sense of immune tolerance, its redundancies, and all its limitations, and for joining us today and giving us all your insights about your career. Thank you.
Absolutely. Thanks for the invite.
My thanks today also go to immunologists who, over the last 100 years, have meticulously followed the biology to advance our understanding of autoimmune diseases. Thank you all for listening to this podcast. ACR Journals on Air is a production of the American College of Rheumatology. Please follow @ACRheum on social media, email us at podcast@rheumatology.org, and rate and subscribe to our podcast at acrjournalsonair.org or wherever you get your podcasts.